As each of these students walk around, their brains are making thousands, if not millions, of movement-related decisions every second. The motor cortex, the part of the brain responsible for voluntary movement, sends thousands of signals per second to activate muscles. Every step, every shift and balance is precisely controlled. The spinal cord handles reflexes, making nearly 100,000 updates per second, allowing for automatic adjustments, like shifting weight to prevent a fall without consciously thinking about it. We understand how complex movement is in humans. But what about simpler organisms? What can we learn from something as simple and small as a cricket? Despite having much simpler nervous systems, crickets still process environmental stimuli and make movement decisions. These decisions may not be as complex as in humans, but they follow patterns that we can observe, quantify, and analyze. Crickets are model organisms, meaning they provide valuable insights into basic behavioral and neural mechanisms that apply across different species, even beyond insects. Considering crickets are relatively simple animals, the behaviors observed in them are more foundational, rudimentary, mostly related to reproduction. However, I noticed female crickets interacting with each other, which could be indicative of more complex social behaviors. I wanted to study how female crickets moved around each other and what it could mean. This raised a new question. How can we quantify these highly nuanced movement behaviors? What if mathematics could be used to see every small movement, every small decision unfold? I began my project in July of 2023 with a very simple experiment. I studied if female crickets showed a preference for another female cricket based on familiarity. My initial experimentation used this rectangular choice chamber, where the focal cricket, whose behavior was being monitored, was allowed to wander between three zones, the one on the far left demonstrating a preference towards another female cricket that it had been familiarized towards, the one on the far right demonstrating a preference towards a cricket it had not been familiarized towards, both respectively in those zones. And the focal cricket was allowed to wander between these three zones, allotted 5 minutes. The total time spent in each of the three zones was recorded, as well as where the cricket was located at each 30-second interval. But over the summer, I found over 100 flaws and places for improvement in my experiment that I wrote down in my notebook. My testing procedure was simple and didn't give insights into many of the very subtle behavioral patterns I was noticing in the female cricket's movement. But by September, school was back in session, so I assumed the project was over. Then, in math and science classes, I started noticing connections. The formulas and concepts I was learning could solve many of the problems I had identified. For example, my initial choice chamber used a Cartesian grid. The problem with this is that the focal cricket could be varying distances from its familiar, but all be measured as the same level of proximity. So when my pre-calculus teacher explained polar coordinates, I thought, hey, that's applicable. Instead of measuring preference based on which side of the chamber the cricket was on, I could define preference zones as regions based on radial distance from each stimuli. So I redesigned my choice chamber to have 2 radial distances demonstrating high blue and low red preference. I also wanted to address the problem that behavioral sciences are prone to subjective interpretation. To make my data collection more efficient, I implemented a computer vision algorithm in Python to track and log their positions in real time. This automation not only saved time, but also reduced observer's bias, ensuring that all movements were recorded with precise accuracy. From there, I was able to add several mathematical models to my algorithm, studying many highly nuanced movement patterns. The results of my study revealed several key insights into the behavior of female crickets, focusing on their preference and movement patterns within the choice chamber. First, by analyzing the crickets' positions and movement data, I found that there were clear areas of preference where the crickets tended to congregate. These were visualized in the form of heat maps, which showed the concentration of crickets in certain parts of the chamber. Specifically, the crickets showed a preference towards the familiar female, with more time spent near that cricket. The next key finding involved the speed of the crickets in relationship to their distance from the familiar cricket. I observed a trend where the focal cricket moved faster as they got further away from their familiar. This suggests that crickets may have been engaging in exploratory behavior as they sought out a new environment, away from the familiar cricket. It also hints at a potential relationship between distance from a familiar and exploratory behavior, where the further a cricket is from the familiar individual, the greater its movement speed, possibly an attempt to explore new areas of the chamber. Another significant result came from the cricket's angular orientation relative to the familiar versus unfamiliar cricket. I used dot product calculations to determine the angle between the body orientation of the focal cricket and the direction towards the familiar cricket. This provided more detailed data on how the cricket positioned themselves in relation to the other females, allowing for a better understanding of social dynamics and preferences at play. I also collected over 1,000 data points, each representing the location of a cricket at a specific time. The data points were used to create various graphs, including ones that showed the cricket's location at specific timestamps and the corresponding behavior. By examining the data in this way, I could identify patterns and correlations between different aspects of the cricket's behavior, such as how their movement speed changed and distance, or how their body orientation varied, depending on their proximity to the familiar cricket. Finally, the methods I used were effective in identifying these patterns, but there were limitations. I had to eliminate 50 control tests from my analysis because the crickets were unknowingly sick at the time, stifling their movement, causing them to simply stay in the neutral zone. This means that some of the earlier conclusions drawn from these tests were not reliable. Despite this, the data from the remaining tests provide clear and actionable insights into the cricket's behavioral patterns. In summary, my analysis of the cricket's behavior revealed distinct patterns of preference and movement, demonstrating that crickets showed a clear preference for familiar individuals, as well as inclination to move faster the farther they were from the familiar. By using mathematical modeling and computational analysis, I was able to extract precise data on these behaviors and gain deeper insights into their social interactions. The patterns in how the crickets move give significant insights into their neural states and how they respond to the external stimuli, particularly in social interactions. This is scalable and could be applied to other animals and species to understand how movement correlates with neural mechanisms. Thank you.